I'm going to introduce to you Dr. Thielen, who's a physician scientist with both clinical and research interests focused on infectious disease, pathogenesis, and immunology, which is really exciting. She's a lifelong gopher, so she completed her undergrad training in microbiology here at the U to University of Washington to do a combined MD PhD program there. After medical school, she returned to the University of Minnesota to complete an internal medicine and pediatrics residency, followed by a fellowship in adult and pediatric infectious diseases. Since joining the pediatrics faculty here in 2020, she's developed an independent research program focused on host-pathogen interactions in the human respiratory tract. She also maintains an active clinical practice in pediatric infectious disease at the Sonic Children's Hospital. So with that, please join me in giving a warm welcome.
Thank you so much today. With dummy pop. So great. This is my go-to this. We have some lab gatherings. It's always nice to have some souvenirs. So yeah, so thank you so much for the invitation. I'm gonna skip to my slides to kind of put my intro. So just to start with some disclosures about funding. The things related, I do get funding from CDC in order to do the studies that I'm going to tell you about today. So just as by way of introduction, I'm going to start with the lab mission and sort of kind of tie that into like the School of Public Health. So I would say it's a three-part. So what we do is three-part goals. One is really focused on respiratory health of children and really thinking about how we develop new tools that are going to improve the health of children globally. Two is like an educational mission. So I really been committed to training, and we have everyone from high school students to clinical fellows that represent in every lab. And we really benefit from the diversity of experiences for our science. And then finally, another big part of what we do is thinking about how we communicate more effectively about science, and that involves trying to find ways of soliciting the input on design of our studies from community members, and also how do we share that back and sort of make participation in studies sort of approachable for people across the state. And so I took this from the School of Public Health website, so I think there's very much a values alignment in sort of the work that I do and what is, the values of the School of Public Health. And in fact, in Seattle, when I started, I was actually based at the School of Public Health there. So it's very nice to kind of build some of those relationships here. So and then by segueing into education, often the credits get put at the end and everybody's tired. And so I really think it's important to like acknowledge the people doing the work so you can see it. So these are you can see spanning undergrads up to clinical fellows, and both here and in one of our study sites in Uganda. And then we have a really strong pediatric research support community, which I think is a unique aspect of the work we do, is we're really specialists who are thinking about how do we engage children in research and awareness, respective of their autonomy and sort of needs and developmental stage, and generate the kind of data that's going to improve the health and sort of allow us to have evidence to guide our practice and clinical care. And so as Lizzie mentioned, I'm a physician scientist, so thinking about this bench to bedside transition. So my bench is at the primarily at the University of Minnesota right now. My fellowship, I worked a lot at downtown St. Paul at the public health lab for our state health department, and I still have very active research collaborations with them. And then my clinical practice, as you mentioned, is at the Sonic Children's Hospital, which is just right around this neighborhood on West Main. So to start about what we do, we really focus on lower respiratory tract infections. So why is that important? So if you look at WHO data, it's actually now the top communicable cause of death globally. So it's right here at number 4. So you have non-communicable diseases like heart disease, COPD, and stroke that have taken over the top positions, but really it comes in as the top infectious cause of death. And there's been pretty small movement on that in the last, in this 10-year interval that they looked. And so those high-level statistics are really just sort of the tip of the iceberg of like delving into thinking about how do you start approaching the kind of big problem. And so the way that I come at it is thinking, well, what are the actual pathogens that are causing that lower respiratory tract disease? And the evidence we have says that viruses are actually really, really important for that morbidity and mortality. So this is a study, kind of a seminal study in epidemiology of pneumonia called the EPIC study. And there were two arms. There's a pediatric group and then an adult group. And they looked at a cohort of hospitalized patients and asked what types of pathogens are we finding in patients with pneumonia. And so on the x-axis here, you can see, maybe. No. So on the x-axis, you can see different age brackets. So the all children and then different subdivisions. And the height of the bars is related to what type of pathogen was found. And so what I'm keying in on is the dark blue is one viral pathogen only, is about a half of all pneumonia has one viral pathogen detected. And then usually the second most abundant group is a viral co-detection. And then you kind of move up to get bacterial viral co-infections. So it looks, to the extent where we can identify a pathogen, viral pathogens seem to be quite common. And when you look at what specific viral pathogens are causing those, what you see is a group, all of these are viruses. So we have things like respiratory syncytial virus, human rhinovirus, human metapneumovirus, adenovirus, parainfluenza virus, influenza, and coronaviruses are kind of making up that burden of viral infections. And it's somewhat similar picture, a little nuance on the adult side. So when you look at what causes pneumonia in adults, the pie chart shows how often a pathogen is identified. And you can see there's a larger portion where we don't have a single causal pathogen identified. But in cases where we did identify a pathogen, it's viral, still the most commonly found. And you see some of the same players. So rhinovirus, influenza, human metapneumovirus, RSV, paraflu. So same suspects are coming up in the adult cohort as well. And so one of the sort of sticky problems that has like been something I've been thinking about for years on this is like why do we see such a spectrum of disease? So I think we often think about viral infections in these mild, outpatient, not that big a deal nuisance infections. And so how do we think about this problem? Why does that sometimes we have this information that it's a cause of death in a lot of people potentially, and yet also know that there's this huge burden that's not causing a lot of symptoms. So I kind of think of it as this volume now. So if we start here, we've got some asymptomatic from mild infections, and then sometimes it goes up to hospitalization and death. And how do we think about ways to sort of understand some of the drivers that are responsible for the biology? This is basically the question that I'm thinking about. And then I'm going to go through kind of a windy the thinking of how some of the ways that I thought about addressing some of these questions. So, my training in Seattle came out of being in the first lab. So I trained as a virologist, sort of a classic laboratory virologist. And so in that first, my sort of default position on this thing, the virus must be the culprit. So let's see and look at the cases where we think it might be a virus. And so this is kind of flashback to like 2016. And I say that just to put you in the mind space, because we've had so much sequencing data that's come out in the last 4 years. We have to kind of like reset your a little bit to where the field was at that point, and people didn't have next strain. We was not looking at next strain to see the COVID circulation. Like genome sequencing was not a thing that was like so whatever as it is. So, I kind of take this point of like, could we use comparative virology to understand some of these differences? And so could we take viruses that are sort of closely related and compare them and how they interact with the cellular immunity to try to understand what was maybe driving some of this difference. And the kind of case example that this is my first fellowship project done at the health department with Ruth Linfield, was looking at influenza C. So influenza C is this virus that's been known for decades. The first isolates were cultured in like the 1940s, so it's been around toxicology for a while. But almost nobody knows anything about it because there's no commercial tests. Like nobody is looking for this. And there's relatively sparse epidemiology in the US. Just from a virological standpoint, I promise there won't be too much virology here, just a little smattering to kind of teach you a little bit of virology. So, this is a main virus. It's DNA, or it's a genetic material is carried in segments. And in typical influenza A and B, which you hear about as causing the big pandemics, this one has an extra, a fewer gene segment. It has this surface glycoprotein called HEF, which is analogous to like the H and N types. If you've heard about influenza kind of typing, it's sort of the equivalent in the influenza C world. And so what was known about this, and again, these studies go back a little ways because this has been really studied mostly clinically for in very limited cases. So it was known that this was detected in individuals who are presenting with both upper and lower respiratory illness. In the sort of bad or good old days of human experimentation in the 1960s, a bunch of healthy volunteers got infected with this virus. And it turned out it produced an illness, but it was a pretty mild upper respiratory illness. And looking at seroprevalence studies, it looked like mostly everybody around the world has been exposed to this virus at some point in early childhood. And there wasn't a lot of active surveillance, like virological surveillance in the United States. And so what was known is that there seemed to be different lineages of this virus that circulating in this based on the HEF gene sequencing. And there seemed to be like waves that would come through. So you'd have one type circulating for a while and then it would get displaced by another lineage. And this is notably not included in any of our vaccines. So our vaccines only have influenza A and B. And the typical most common therapy that we use in clinical practice, oseltamivir, is not active against this virus. We don't have any treatment tools. And then to add to the difficulty, it's difficult to grow in culture, not impossible, but difficult. And we didn't have molecular tests. So we were sort of flying under the radar of what we knew. And so for this project, we wanted to kind of fill in some of the gaps in an understanding of the biology of this virus. And so to do this, we really took advantage of what is a really strong infrastructure we have in Minnesota for respiratory pathogen surveillance. And this is largely thanks to Ruth Linfield, who is our state epidemiologist and sort of my research mentor in this. So we have three, just to orient you to what kind of information is available. We have three sources of samples for this study that I'll tell you about. The first is a project called SARI, or Severe Acute Respiratory Illness Surveillance. And this is hospitalized patients with respiratory illness. So a couple of sentinel hospital sites at Regions, Children's Hospital and Clinics of Minnesota, and for this. Then we have an outpatient surveillance cohort, and this is varied over time, but involves sort of outpatient clinic sites in patients with an influenza-like illness. So typically like fever and respiratory symptoms, and they get a collection of specimens with a little bit of data associated with them and such. And then sort of on the opposite end of disease severity is this unexplained critical illness project. And so that's a system whereby healthcare providers across the state can submit samples to the health department for like advanced detection in cases where somebody was previously well, suddenly becomes very ill, and the routine clinical testing does not identify a cause. So this is sort of a rich repository of samples over multiple years that the health department has to potentially go back to and do testing. And so as part of this study, we performed influenza C testing on all respiratory surveillance samples across that about a 3.5-year period. So in the end, we ended up testing about 10,000 hospitalized patients, and we had about 2,000 in this outpatient group. You can see that the demographics are pretty different. So SARI is largely skewed toward the younger age range, and that's largely driven by the fact that children submit so many samples. And I should say these are all waste clinical samples. So the only way that a test gets generated is if the lab had a clinical test sent for some kind of respiratory test, and then there was waste material left over. So if somebody looked at the patient and said, this is probably a virus, we're just going to treat you supportively, we would never see that because they're not going to get a swab sample for testing. And so for all of these individuals, we have clinical characteristics extracted from in a case report form, and then we had waste specimens, which is often a nasal wash or a nasopharyngeal swab, in some cases a BAL, a bronchoalveolar lavage. And then the health department at that time was routinely doing PCR, multiplex PCR testing for 22 common respiratory pathogens. So we had all those data. And then at the same time, we added on a real-time RT-PCR for influenza C, and then did some half gene sequencing to kind of look at the genetic diversity of the virus. And all in all, we ended up identifying 70 cases of influenza C that really were a wide range of ages, but a median of 20 months. And we found a lot more in the hospitalized group, but that also was in the group where we just had a lot more sampling. And then in terms of co-pathogens, we often in virology think about, well, if something is present, is it truly causing the disease, or might it be a bystander and something else is actually pathogenic? And so we really wanted to look at, well, how often are we finding influenza C alone without any other potential explanation for symptoms? And it was about a third. We actually found it as the sole pathogen detected. So looking at the time period here, the samples are really distributed kind of across this time period. But interestingly, the whole first season of this, we didn't see any cases. And so it kind of like, I think people were a little nervous. They're like, we're not actually going to find anything. This is going to be a totally dud study. And then we had the season where we just had like an explosion of cases. You can see like in the 2014 to 2015 season. And then it kind of petered out a bit. And so this kind of pattern of some of the respiratory viruses do circulate with a periodicity of more than a year. So it is quite common that some viruses will do 2-year cycles. And it's hard to say without more data, but I think that was sort of a hypothesis here, that we have to catch one of those peaks, and then it was a different annual periodicity. When we looked at the ages, so we saw really the most positive specimens were in that 6-month, 2-year age range, which is really where we see a lot of our circulation. But we did see cases across the age range, and we saw even adults coming up as a single pathogen. And then in terms of the lineages, so I'm not going to go in super in detail of the molecular epi here. I'll talk about it later. But basically, we were able to sequence the surface protein and then compare it across other viruses to see what are the closest relatives. And what we ended up seeing is that there's 2 co-circulating lineages in Minnesota, one called Sao Paulo and called Kanagawa. And basically, we did see this. I think these are small numbers, and I think I would say more hypothesis generating. But we did see a little bit of a shift towards finding the Kanagawa strain more commonly in the hospitalized patients. I think kind of an interesting observation, but I think given the small numbers, it's hard to know exactly how much to make about. But in summary with this project, what were my takeaways in thinking about epidemiology? One I will not talk about, which is how do we take these findings and test them in a model system. So I'll say that for a separate conversation. But I think that was one of the things, is how do we take these epidemiological observations and sort of get at more causal issues and mechanistic. And I think that's where my lab also works in more of a cell culture model system. But we did feel like we could say that influenza has a seasonality, and there's substantial year-to-year variation in how many cases we see. It definitely seems to be predominantly found in children, but certainly not limited to children. We definitely see cases in adults as well. And I think what we kind of added to the literature is there was a perception, at least, that flu C was really just a mild, not inconsequential disease. And we did actually, in this case, we were able to find it in patients who seemed pretty severely ill enough to be hospitalized, and in patients that didn't have other potential explanations for their disease. So I think it sort of raises the possibility, at least, that this may be a little bit more of an important human pathogen than maybe was previously appreciated. And so we had some suggestions in the sequencing data that different types of the virus might have differences in pathogenicity, but I think it's something that still merits further investigation. I think there were also some really important limitations that I learned about through this study that have informed my subsequent work. So in this field in general, we tend to oversample in the severely ill patients. So we had a lot more cases of hospitalized patients, and we know far, far less about mild disease, and even more so the patients that aren't even symptomatic enough to come to medical attention. And so I think there's just tip of the iceberg really of what we're sampling. And I think the conclusions that we draw are skewed by the fact that we're looking at a very select subset of patients. No testing asymptomatic patients. And then I think the other piece that's hard to pull out of this type of study is that it's hard to know how much of it is of the potential differences with strains that is dependent on intrinsic pathogenicity, like how much damage the virus caused, versus what is the past immunological experience of the virus. So if, for example, Sao Paulo was a virus that had been circulating previously and people already had some immunity, that might look very different than if Kanagawa was like a newly introduced lineage that people was more novel to the population. And so I think studies that sort of integrate past exposure history and virological determinants are things important things. And I would say, I'm happy if people have questions along the way, I'm actually happy to stop and take questions, so feel free to raise your hand and stuff.
Just thinking, like, how would you get at that sort of past exposure? But it'd be years and years, you're gonna be,
Yeah.
the front swab is not. You.
Right, so I think, and I'll get to that, I think the answer for that is longitudinal studies. Where you have some data over time and you do start to generate people, individual specific past community, and people are, they're a huge amount of work, but people are doing those kinds of studies to look at these questions of repeat exposures. That. Yeah.
I could see it. I just out of curiosity,
Yeah.
from your body, see,
Yeah.
it looked like rhinovirus. There was a lot of in Seattle.
Yeah, I'm hearing about this, we're actually about to together.
So she focused on rhinovirus, and maybe it's just that she did most of her research. I thought actually. My understanding was that lower respiratory infection immune. Is that the case, or does it also help people?
That was a really sort of incompletely answered question. So the thing that's interesting about the virus is it is an entity. It is actually an extremely heterogeneous, genetically diverse group of viruses. And they all, when you do the clinical testing, you get one answer. Like, is it rhinovirus? And then actually the first part of it actually cross-reacts with enteroviruses, which are another related group. And so the ability, I think it's hard. I think people need to do more studies looking at strain specific detections, because I suspect that there's heterogeneity in there. And it is known for other, not like lower respiratory tract disease, but diseases like acute flaccid colitis, which is very clearly linked to it, not perfectly, but very strongly associated with enterovirus D6E. And so I think, and there's certain strains of rhinoviruses and enteroviruses that are associated with specific disease manifestations. And so I think a lot more people need to do thinking about what maybe a rhinovirus is just not entity, it's actually multiple things. So I think it's really tricky though, because it does turn up in about the same frequency and these symptomatic rates that people as severely ill people. You know, some do find it in people who are like don't have any other clear explanation. So that's a really fascinating kind of virus, and one that I think it needs a lot more studied.
When you were looking, I know you looked at co-infection with Lucy. Did it seem like there was anything co-circulating like seasonally, not necessarily within most, but
Like that certain viruses are found more frequently with it. So we looked at that and compared like overall in the population, what were the frequencies of different viruses versus what was found in co-infection, and actually pretty well tracked. Like you might expect based on probabilistically what would turn out in a co-infection. But I think that's interesting because we do see other viruses like para-influenza, like certain strains alternate, like you see one year and then a different one the next year, and they tend, if you look at that because over time, they tend to oscillate. And so I think there may be aspects of that, but we weren't able to pull that out here. And so I think, this is sort of wrapping up this project and getting it out. It was sort of happenstance that something like new, interesting that it came up. And this kind of like the benefit of being a clinician is you see something and you have the tools of scientists, you like to this question. So I was doing a clinical rotation at the Pathogen, and one of my colleagues, he said, "Hey, Beth, we're seeing this like a lot of these cases of RSVB. It was summer, and people know this. Like that's unusual. Like we see a lot of RSV in the winter. It's like December, March to March kind of. So to see it in like June and July is very atypical. And then RSVB, so we can distinguish whether it's RSVA or RSVB, and they're all RSVB. And so is there anything we can do, the health department to try to like sort this out? And so again, I'm back to what we have in the sample banks and our existing infrastructure. And it turns out that respiratory syncytial virus is a reportable disease in the state of Minnesota. So this is the example of like knowing what things have to get reported to the health department. So you're going to have better capture of events that are reportable diseases. And actually RSV is part of this sentinel surveillance. And at the time, it was required that all hospitalized cases of RSV in the 7-county metro get reported to the health department. And it's now expanding, in the process of expanding to statewide to have this kind of, metro cases. And over multiple years, so we were able to do a look back at what we were seeing. And it's a little bit, maybe less impressive here, but knowing that typically this is the time period when we first got the call. And compared to previous seasons, there's a small number, but there's definitely more cases than we've really seen in the summer times over the previous, going back to 2013. Oops. And it also, we saw the following fall, probably quite a bit more cases coming up, including cases among adults. So the reddish color is the over 18 population. So we're seeing more cases. And so, my question, this seemed like a change in epidemiology kind of suddenly. And so it seemed like potentially a place where we might get information from looking at the virus itself. And so to orient to RSV, so this is an RNA virus. It has a 15,000 base pair genome, and that genome encodes all these different proteins. And then the potentially the interesting ones that people may be familiar with are these surface proteins, so FG and F. And that's where typically we think neutralizing antibodies are directed. So if you think of like the H1N1 kind of typing for flu, it's sort of the equivalent for RSV. So if you're thinking about a new virus, potentially new immunological epitopes, that seems like probably a place you might. And so I'm going to take a pause at this moment, since I know people aren't viral epidemiologists, and sort of just kind of go over a few concepts here of viral epidemiology. So, genetic diversity, where does that come from? And so typically when viruses are replicating themselves, they do that with a protein called a polymerase. And that just inevitably makes copying errors when it's making new cycles of virus. And RNA viruses like influenza and RSV have a higher error rate, so they're just prone to making more errors than our cells or other like DNA viruses typically. And so they also replicate really fast. So the amount of viruses that get made over, even just a period of days is huge. So if you look at a rate of error production and just multiple cycles of replication, it introduces a lot of variation in the virus that can be tracked over time. And it's a way of sort of looking at relatedness of viruses. So when I use the term whole genome sequencing, I'm talking about looking at the diversity of the whole viral genome. Frequently, historically, because of our technology, we're limiting sequencing to small areas, but actually there's a lot more real estate of the virus that you can look at for potential changes and interesting sort of effects if you look at the whole genome. And to introduce another term, often what we're seeing are these single nucleotide polymorphisms, so single base pair changes in the sequence. And a typical readout looks something like this, where you see, the predominant, like in this purple, most of these have a G at this first position, but here's one virus, one pathogen that has A. And then, here you have multiple of the sequences that actually have a substitution to a T at this position. And so there's sort of a spectrum you can think about of things that are highly variable. There's a lot of different options that could be at that position, or you have like conserved, which means that everything is identical at that position. And then you can sort of use software programs to sort of create a likely relationship or phylogenetic tree based on the pattern of those changes. And so you look for things, you kind of cluster together things that are most similar. So here, A and B both share these 3 unique changes. And so we think that they're probably most closely related. And here we see C has, 2 of these changes in common, but it has something unique, and then it doesn't have this green one. So it's sort of related, but like less so than the A and B are to each other. And then you find other things where like this one has this whole different red one here, but it has these 2 other, changes. And so you can basically look at this constellation of changes and try to infer relatedness amongst different viruses. And so this when I kind of talk about molecular epidemiology and viral speak, this is kind of looking at building relationships from genome sequencing. And it also, as a biologist, it can run some hypotheses about what role different parts of the genome might be doing. And so in general, you can look across the genome. This is an example from Anne Tollenberg looking at rhinovirus sequencing. But what you can see is that overall, the positions, if you compare different rhinoviruses, are about 63% identical. But there's clearly points where there's a lot of diversity. So this is like like very little similarity amongst different strains. And then you see other areas that have like almost 100% conservation. And so, like at a first pass, you can think of areas that have high conservation, maybe are likely to have an important function, like they're doing something critical and they will not tolerate change. Versus something where there's a lot of diversity, that might be something that's important for immune recognition. And so there's a selective advantage to the virus changing at that position to evade immunity. So this is some of the kind of information that we take out by looking at these types of sequences. The other concept that I want to introduce is sort of the sequencing revolution. And so for most of molecular, like the history of virology, sequencing was done where we looked at like one molecule at a time through Sanger sequencing. And I think this field has really exploded as next generation sequencing has really taken off. So we're now able to look at millions of different sequences at the same time, and that really like expands the scope of what we can do to look at viruses. And so I will go through this kind of quickly just for people who might be interested in the details. So we got the sequences, we ultimately got sequences from all of these hospitalized patients at Hennepin. We had a bunch of comparator sequences. We went back into the freezer archives and pulled old RSV sequences from Minnesota over different time periods. And we prospectively collected some additional specimens from other hospitals like Children's. And we did some like biochemical pre-processing, and I basically collected the RNA from the virus to use two different parallel approaches, and then prepared the library and did sequencing on the Illumina MiSeq instrument at the health department. And just in summary, this is all published, so I just kind of give you the punchline, which is that we found that the sequences that were starting to crop up in 2017 all seem to cluster, the majority of them seem to cluster together, and they clustered distinct from some of the sequences that were circulating in Minnesota the year before, and they were also distinct from a number of reference sequences that we pulled from GenBank, which is sort of an international repository for genomic sequences. And one interesting commonality was that we saw this one shared point mutation leading to an amino acid substitution. And it sort of, it had occurred not for these candidate surface proteins, but actually a third less well studied protein called SH. And is also expressed on the surface of the virus. And this is showing sort of how that particular single sequence variant sort of expanded into the population. So, and when we look back at some of the sequences we had in the archives from 2016, we didn't see that particular variant at all until early 2017, and it started as a minority population. And then by that summertime, we saw that break happening, we see complete, like basically complete takeover of that sequence variant in the population. And so lessons learned from this. So I think, we would have totally missed this if we had limited ourselves to just very limited subset of the sequencing of the virus. I think it really provided for that a case example where that additional information across the virus was potentially useful to understanding what was happening. And I think it becomes a lot clearer when you have more samples over time. And I think this was the value of having all these archived specimens. We were able to go back to and say, hey, find out that this actually was different from previous populations. And I think it also highlights the importance of having clinical data. Like it's great to kind of look at the epidemiology and isolate, see what strains are coming in, but I think the potential implications become a lot more significant when you're able to pair that with knowing that the clinical data was that we're seeing this change in the circulation pattern of viruses. And so that sort of brings me up to 2020. And at this point, this was when I sort of was like ready to like fly out of the nest and do my own thing. And if you'd asked me at the time, this was like really more what it looked like. The person who had been working with the lab got taken over for COVID testing and had been in a different space. I couldn't order anything, couldn't hire anything, couldn't be in the same space as everyone. So this was like a huge excitement, I think I'll say today. It did not feel that way. I think we've come past that. And but I had a lot of like sitting alone in this thing. And I'm thinking like what's next, having this existential crisis and thinking about all the things that we know that like age and like comorbidities are factors. And I'm like, where can I go take this that is different from those things that we have known about? And so, I'm a sort of dyed-in-the-wool microbiologist, and I love viruses most, but I also love bacteria too and other microbes. Choose. The supporter of the trip. So, I was kind of interested in this like microbiome. It's just like this wave of like information coming out about all the things that the normal microbiota do, and including like influencing pneumonia. And I became very intrigued by some work from Europe, from Debbie Bobert's lab, looking at the respiratory microbiota, how it develops in the course of early childhood, and how it seems to be sort of correlative with outcomes like respiratory, like viral type or infectious pneumonia and respiratory illness. And basically, this is a review, but it summarizes a sort of body of work that generally has established there's different patterns of nasal microbiota, and that certain types of microbes tend to correlate with relative few respiratory infections, and other types correlate with having more infections. And so this is kind of in the know, as you can see, there's different patterns as you go along the respiratory tract. So it's important to know that the respiratory system is not one monolithic thing. It's actually a lot of different ecosystems that have sort of different microbiota. We've been focusing mainly on the nasal microbiota because it's easy to see, actually, in the reason. And then in this previous work, though, she was really looking at the development and correlating with known epidemiological risk factors like mode of delivery and breast milk versus formula consumption. And so less work specifically on viral, like specific named viral pathogens. And what was kind of clear is that also from other cross-sectional studies is that there's actually dysbiosis. So like sort of some of these abnormal types of bacteria were associated with acute infection. But a lot of those studies were done as single points of time, and so it's hard to know, like, were those pre-existing and the virus took advantage of that, or these consequences of the virus. So they're really big, a lot of these things are really big chicken and egg. And so, we wanted to, that seemed like they were suggestive of in both human studies and also some experimental work in mice, saying that this is potentially actually a sort of may have a causal sort of role in changing susceptibility to respiratory infection. And so formulated this idea, which I borrowed with compliments to Carrie Buckington, who did this Big Love study, which is a longitudinal study in viruses in Salt Lake City. And I thought, let's add to that, and let's take advantage of some of these new technologies that are coming out in terms of next generation sequencing. And that from that was MINNE-LOVE. So this is like a tribute to Big Love. And so what's the study design here? So we're going to look specifically at children under 5. And the reason for this was really practical because we knew as from the previous data that they're just like virus machines. Like there's just if you want to accrue a like there's no better population than children under 5. And then we did kind of thinking forward, I'm interested in other things, and so post-genetics is a little side interest, and so we do actually have buccal swabs in these patients to go back to the future and actually look at genetic things should it become interesting. Then we did longitudinal sampling over time to compare sort of how the microbiome changes in response to events. So for all these participants, we're collecting weekly electronic symptom questionnaires, and then the family collects a nasal swab, and these are specially designed for microbiome stability. So the idea is that you will collect it, it will sort of freeze the microbiome, and you should have shifts in nailing conditions. They're stable across a wide temperature range. They tolerate freezing like Minnesota winters. These are in Ontario, so like it's really a great system for here. And then the initial plan is that we were going to do this it's a kind of a metagenomics class approach. So generally broad sequencing, but we're using something called hybrid capture to really sort of fish out things of interest and really focusing on viral infections. And then we're also going to do 16S gene sequencing to look at the nasal microbiome position. So I'm glad Ellen is here to see our progress because we've been a lot of knowledge of the state fair in terms of our recruiting. So, and this is like the first iteration of our talk. Was at the county. This is one of the county fairs we went to. But we've been working 3 years now. The first year was sort of a small test year. We had 7 participants and we got 130 specimens back for 71% return rate. We did some method improvement, and in year 2, we sort of doubled our participant population, and our specimen numbers, and we have 91% return rate. And then this year we're actively enrolling. We have 32 participants who are in so far, and 260 specimens back. So still going. We should finish out in June, I think. And so I will say, like the whole title of the talk was really reference to this as we're at the fairs. There's milk stations, like those kind of to the cute. And then when you asked me for the talk title, I was like really serious and like taken seriously as an academic. And he's like, just go with the guts now. It's a placeholder. And then I was walking around and like one of our new pulmonologists saw me and she's like, hey, you're the snot person. I was like, well, I guess I can't change it now. I'm gonna lean into it. So that's we're now the snot. And we actually did have a really fun time with this is an otolaryngologist we did sort of a co-branded experience here where they were doing some ears of your nose, and I will tell you, I had no idea that we're so motivated by getting a chance to look inside their own ears. Crazy. But yeah, there's a lot of like community interest.
They love your guys' stuff. State. Here we go. You guys are there with your like ears and like people in, and it's just amazing.
Yeah,
Very interactive.
yeah. So here's some of the actual data, and so this has been slow coming because we were actually originally doing this with the Genome Center, and we're currently in the process of transitioning it over the workflow over. So these are 4 participants. This is an example of the data, and I'm focusing on microbiome data. So each of these blocks of 4 is a unique person, a unique child. These 2 are siblings actually in the same household. And then each of the single columns is 1 week under study. And then we looked at through the 16S sequencing, what genetic bacteria are in the nasal swabs. And so I think what you can see just from the first pass is there's a lot of there's differences between individuals, but within individuals, there seems to be a lot of similarity over time. So like this one looks kind of remarkably like itself through all the weeks. And it looks very different from like this participant. You also see some like big shifts in the microbiome. And so you see these points like here, where you suddenly see encroachment of this green organism that kind of blooms and then goes away. And so that is Haemophilus. So people are medically know about this, like Haemophilus influenzae is one of these big respiratory pathogens. And so we do see, we've seen some interesting things where there's seems to be, big shifts in the microbiome. And then if you look at a PCOA physical component kind of analysis of these, it sort of shows in a different way, but I think you can tell visually, which is samples from an individual person that tend to cluster together. So each of these circles is a color coded for one of the individuals. These are those 2 on the far right, and the siblings. And you can see, I think that also has some validity that the siblings who are in the same household actually are more like each other than the other participants. So these are the ones we've got the data back on, but we have like tons of specimens archived that we're hoping to kind of, now that we got everything seems to be working, kind of plow through and so we add a lot more cases to our counts. This is the viral data. And so each of these is again the same 4 participants, weeks 1 through 26, and then which viruses were detected, and the weeks of detection are highlighted in yellow. And so what I'd first kind of point out is there's a couple of viruses that we don't have to regularly test for, but are the herpes viruses. So herpes HH3/6, CMV, and ADV. These are viruses that are DNA viruses that are known to cause latent infections with persistent shedding. And so we know that once they're in the body, they never go away, and you can detect detect virus intermittently over time. And this is exactly what we're seeing. So some of this participant basically has a shedding HH3/6 at a low level throughout the study interval. Probably not sick from this, but has a potential for transmission. And so similar with CMV and EDV, that we see that the viruses that have the long periods of detection, I think fits with what we know of the epidemiology of herpes viruses. We also see a bunch of these short-term bursts of different viruses, and there's the whole range of viruses. We see rhinoviruses, we actually captured an event of influenza C, even though it's 4 patients, we happened to capture one influenza C infection, which is really cool. But But I think this really shows we can really detect a lot of viruses. I think we see what we would probably expect, which is when we see detection, we do see it sometimes across multiple weeks, and they tend to be contiguous weeks. Yes.
Do you know, if any of these four were sick during the period? Or you get to that?
Okay, oh shoot, sorry, we do, sorry, those slides. So we also have paired symptom data, and I will tell you that like congestion is like a constant stage. Like over half the weeks of all the participants have congestion reported. And so and there's like less fever, but see, there's kind of the first pass, like a fairly like dissociation between, we see so many viral infections coming up, and it's just like a sea of like constant symptoms. So like it's in this age, I mean, it's just going to be clearer when we start sampling adults that you'll see more clearly like symptoms associated with illness. But yeah, for these young ones, it's just so I think, the first big move here was like this parent collected remote sampling is feasible. And I think this really is a great tool to expand our ability to study mild and asymptomatic infection. The longitudinal sampling can kind of minimize the noise. And so I think when we look at microbiome studies, often they're comparing big groups, and we aren't able to sometimes say what's like an individual idiosyncratic difference versus what is potentially related to the exposure of interest. And so I think the fact that we can use people as their own controls, it like will help us to see what, when we have a new event coming in, a new virus, we can look before and after, and maybe it maybe be a little bit easier to see what kind of effects are correlating there. And then I think this kind of using this kind of sequencing bank-based platform really lets us think about adding on additional analyses. So looking at human genomics, looking at host transcriptomics, other things that may be informative. And I think we did really like that the participant engagement helps. And we have done some things with our RD that allows us to share reports back at an individual level to participants. They're not clinical testing, so we're not telling them in real time that you have RSV, but they can have access to, for their own individual data, what they have over the course of the study, which I think this group is largely healthcare worker parents. But that, for subset of people, that's like highly motivating. And so now that we've got this, like these cool tools, kind of like let's broaden the landscape and look at like what else, what are we gonna tech next? And so the big project that we're getting ready to launch is really how do we take these tools and look more broadly across the population? And how do we think about using this as a tool to try to expand the reach in terms of types of participants that we're including? And so I think that from COVID-19, I think everyone is probably very familiar with the fact that there's really massive disparities across a lot of axes in terms of things like hospitalization, access to testing, access to vaccination, things like that. And I think, having the ability to bring testing out into the community and make it easy for people, I think is something that offers an opportunity to expand who we include. And so as I was sharing some of these data with people at the health department, we had this funding opportunity that came up for this new 5-year sort of endeavor called the Pathogen Genomics Centers of Excellence. And we applied it I applied it with the colleagues at the health department for this grant, and we ended up being awarded it. And now we're one of 5 centers across the country that are really interested in kind of doing more work around certain health, like epidemiology and in the community. And so this is sort of the mission of this grant, to foster and improve innovation and technical capacity in pathogen genomics, molecular epi, and bioinformatics to better prevent, control, and respond to microbial threats of public health importance. And so what we propose as our sub-project within this larger network is to really double down on like how do we get more people, diversify our population that we're including. And so we're looking to enroll 500 households from across the state. And so we're calling this MINNE-LOVE 2. And again, we're going to have a buccal swab for the future. But we're going to keep this pattern of doing weekly symptom surveys, and then provide households with swabs to collect samples when they're symptomatic, if someone's symptomatic. And then we're going to do respiratory pathogen identification. And instead of doing this sort of short read technology, which is through a genome center, it's very centralized, it's very expensive, it's very much designed for large numbers. We're in the process of bringing it into my lab, so we're using an instrument that's going to hopefully help us do this in more real time, do small batches, and kind of shorten the turnaround time to getting data back so that we can actually report it back to participants. And we're really being like thoughtful and deliberative about how we do dedicated outreach to different communities that have been historically left out of a lot of these studies and build relationships. And so that's something we're spending a lot of time. We are hopefully going to be enrolling for this soon. So right now this link takes you to our website, but once we get the bot or pre-screen there, anybody who's interested in participating can indicate interest, and then our coordinator will reach out. So in the last few minutes, I want to share some data from our global. So let's take this even bigger. Where is this going to be useful for epidemiology globally? And so I will highlight the importance of this, that we told you that overall infections are the fourth leading cause of death globally. But if you actually look at where you break that up by income state, you look to the high income countries, it's actually number 6. But if you go into low income countries, it actually moves up to number 2. And so these diseases are disproportionately affecting places with resource constraints. And if you specifically look at children with pneumonia, lower respiratory tract infections kill over 650,000 children a year. And it is skewed, you can see in the US, it's blue, it's like very low rates, 0 to 4 per 100,000. But if you look at places like sub-Saharan Africa, where I worked in Uganda, these rates are like extremely high, much higher. And so, you know, taking this like large concern, how can we start sort of thinking about applying some of these techniques? And I met a colleague who's actually a oncologist, and I will say that's an emerging field of being able to treat cancer in low resource settings. And that, of course, these children are immunocompromised. Infections are a big problem. So they put kids at risk. And infections that may not be harmful necessarily even to well kids are really problematic when you're immunocompromised. And so we're looking at this condition called febrile neutropenia, which is when children are receiving chemotherapy, the chemotherapy knocks out their white blood cells for a time, and they're like lose the ability to control a lot of infections. In terms of when fevers are often presumed as a sign of infection, and using the sort of best locally available technology, which is blood cultures, we are only able to identify a cause of that fever like 14% of the time at this center. So this is data from a colleague in microbiology, Maggie Lubama. And we know from our US data that like immunocompromised children will have a lot of opportunistic and different pathogens, including viruses. And so we sort of had this hypothesis that respiratory viruses were probably an important cause of fevers. And though we at this point don't necessarily have dedicated treatment, knowing that there was a potential cause of fever has the potential benefit of being able to narrow antibiotics, not to pile on a bunch of additional coverage when you're just guessing at what might be causing the fever. And so we had this idea that they had such limited microbiology capacity there, but we thought the sequencing kind of approaches might be nice because it would allow potential diagnosis of a lot of different pathogens with a single tool and not having a lot of sort of one-off thing that sort of unitasker kind of diagnostics. So this project was taught by Dr. Benigna Navarro, who did this as part of the project. So she was enrolling children admitted for chemotherapy at the Uganda Cancer Institute. We wanted to have both a febrile group and sort of asymptomatic control group, so kids who are coming in with neutropenia, but not the fever. And then we collected a clinical questionnaire that had some data about their oncological history, what symptoms they're having, routine labs, and then everybody got an admission nasal swab. And we used the same diagnostic platform that I just showed you. It was right here, did it there. And we actually did it in country. So we have a sequencing facility that got put in during COVID. And we said, you know, I know you're sequencing COVID, but could you use this instrument for another purpose and test for it? So they got this set up for us. And I think the money slide is this. So in terms of detection, I'm sorry, the black, I have to retool this for better visualization, but you can basically see the blue is the number of kids with virus identified. So you see about half of the kids that were febrile had at least one virus detected, whereas about a quarter of the asymptomatic kids did. So I think this fits with what's known in the literature. There's just a lot of viruses in kids and they may or may not be symptomatic. We do see an enrichment for viral infection in the febrile kids. And then when we looked at what those were, so again, similar to suspects, we saw C-virus, SARS-CoV-2, a whole bunch of different things. And I think we did see, you can see a lot of the classic sort of respiratory viruses like RSV, flu, parainfluenza virus. We only saw kids with fevers. So I think it was really exciting. Because I think it's sort of a proof of concept that we can use sequencing technology to potentially expand diagnostic capacity in places with limited conventional biology. I think Colorado did raise an important limitation, which is the cost and turnaround time issues that are going to limit clinical utility. And so where is using, I mentioned briefly the different sequencing modalities. This is not meant to be a whole detailed talk of sequencing, but in short, what we're using is Illumina, these large instruments. They set up at big sequencing facilities. But there's this other technology which is called long-read sequencing or respiratory sequencing. That's actually been in use for over 10 years in just this kind of environment that are fairly resource constrained. You can see the instrument kind of fits in this like suitcase you can take. These are published data out of the West African in 2014, where they built some of these phylogenetic trees that you saw earlier to try to understand transmission dynamics. It actually was critical for picking up some of this occult sources of transmission, like long-term shedding and genital fluids and things like that, which would lead to these like recrudescences of infection like months after it seemed like the epidemic was under control. And so it really was a nice example of like this like molecular epi or traditional epi investigations. And so this is like a review comparing these two platforms. So Illumina on the left, right, and Sudanapore on the left. And I think the key details is this is like 20 hours, and this could be done in as little as 6 hours, and it can be done with a lot less infrastructure. And it's even being put to use. People are thinking about how to use this for diagnosing. And this is a picture from my lab. We just got our instrument delivered a couple weeks ago, and so this is where we're actively working to bring out some of these techniques. That was my last slide. A ton of people to thank because I really do work with a lot of different people, including stuff that I didn't talk about today. But I really specifically call out the team at the health department. So Ruth, Sean, and Sarah, the co-PIs for this new PGCOE grant, and have been friends and collaborators for a lot of years now, and it's really exciting on this project. And then I specifically call out our MINNE-LOVE cohort, including Alexander and Olivia, who have more or less been with me since the beginning and have just tackled a ton of different tasks. So whatever it's to do, it really wouldn't be possible without their help years. So I will stop there and take additional questions.
Thank you so much. We have time for a couple of questions. You know, people were asking questions throughout too.
So not so much about the science, but about the infrastructure. So just privacy advocates in the state get pretty bent out of shape. Their data being cancer and also around screening and routine spots. So tell me the experience around reporting and having those samples, and do people know it and are they real?
So certainly there are kinds of questions that I think have gotten, and I think to some extent our participants so far have been a little bit of a selected population that people who are a little bit more healthcare sort of involved, I would say, and have less of those concerns. I think we're clear with people that like for most of our reporting back, unless it's like an epidemiological, like an urgent concern, like somebody has we've had measles, for example. We aren't reporting back identified information to the state. Everything is contained within our lab. And so people can get access to their individual reports, but we are not publicly getting reported or getting reported back to the health.
Right.
Yeah.
Like what about, so they have this surveillance thing,
Yeah.
clinical waste is kept.
Mm-hmm.
Do people know?
That's no. I think probably not. Yeah. Yeah. Yep. Yeah. Yeah.
Thanks for that. That was great. So for this sequencing in Africa, just curious, like to what extent you have conversation with people in HIV, because like, in the US, if you suspect drug resistance, you just get a drug resistance test. But in Africa for decades, basically not a thing. The turnaround time, if it exists, would probably be like weeks. So So how realistic is it, do you think? And in what kind of time?
So it's a good question. I agree, like a lot of those, and I think for here, it's all done in the context of a study, so, you know, we have funds to pay for it, which it's still, it is overreach to do this clinically right. But I think, you know, from what I've heard in terms of the Oxford nanopore sequencing, if we can kind of tool and optimize, I think it could be in the cost range of like what is a blood culture and maybe some ancillary testing could be done. And I think if we have, you know, there or in also starting a work in Nigeria, and we have a lot of micro labs that there at the hospital. And so I think with the right level of iteration, like and maybe in a 5-year time frame, we could get this from being a research test to something that they actually could potentially do there and for clinically. And I think it's really variable. So like there's definitely like a lot of patients who pay for basic things that like that, you know, it's all self-pay basically. But there's also people who have some resources, and I think that, you know, we could get this, I think, to a place where it could be. I mean, certainly the multiplex panels are way out, because the commercial cost for those, even if the research use only, is like $150. So like, I think that is out of the range, but I think I could see this kind of maybe getting to $10 or $15 maybe. There's a similar study that looked at doing genomic testing for some of the hemoglobin genes in Tanzania, and they were able to get that down to a cost of $10. So I think that could be in reach.
Okay, one more question and then. Hi, I'm Erica. I'm a PhD student. I also used to work with undercarriage. And this is maybe just a representation of the fact that I don't know anything about. But you have those like 4 patients. First of all, do you expect, like, is that the amount of interpersonal variation that you expect? It's like if you have 100 samples, they're all going to be that varied. And if so, you analyze that data. Like I understand how you can look at it and be like, oh, look, you can do that. But how do you analyze that data across 100 people to come up with something that is like, well, generalist? So it's a big question. No, it's something. So Ron Blackman has been our collaborator, and Sabrina is a PhD student, and they're named to the University of California for this, but they're still actively working with these questions. Like how do we think about tools to provide data over time and make those comparisons? So I would say there are some other examples, but I think this is very much in development of how we think about this. And I think we're going to need to generate have more examples of participants to be able to like fully map out what is that diversity over time. I think broadly speaking, people talk about the word, the sort of type, like there's broad types that have certain components. And I think where the microbiome that I think has struggled for a while is our initial measurements are very specific things like alpha beta diversity, like how many species are there? And I think as a microbiologist, that's really very like high level and almost to the point of not being useful kind of measure. Because I think where the pathology is actually probably the metabolic that these microbes make and how they triggered. Well, the hypothesis we have is that they actually trigger the pathogen recognition receptors through common molecular patterns, and that mediates the immune response. And so I think you need to take a more sophisticated microbiological look at what are the actual component species, what is the metabolic way that they have, and what are they doing to the immune system. I think more to come on. It seems like the field in general going a lot more, and I think also not limiting ourselves to just like general genus level identifications, but actually doing more metagenomics and looking at metabolic pathways and stuff. And I think that's probably going to be the thing that leads to better understanding and making things a little bit more predictable in this. okay, well, we are a little past 11. I just want to say thank you so much, Beth. That was fantastic. I learned a ton of things. I will see all of you next week, hopefully.